Welcome to ASGCT's The Issue Podcast. I'm Jason West, joined by Sanjay D'Souza. We're recording this live from ASGCT's annual meeting in Boston, 2026. Sanjay?
Before we get started, join leading policymakers, industry executives, and scientific stakeholders at ASGCT Policy Summit, a one-day event in Washington, D.C. on Monday, September 14th, 2026. Be there to discuss the most pressing issues in the field and connect with leaders working across regulatory, manufacturing, access, and innovating in CGT. Virtual registration is also available. Visit asgct.org to learn more.
Anyone who's worked in gene editing, in the gene editing field, remembers two landmark science papers that helped define the modern CRISPR era. The first, published in 2012 by Emmanuel Charpentier and Jennifer Doudna, demonstrated how CRISPR-Cas9 could be programmed for targeted DNA cleavage. And the second, published in early 2013 from the lab of Hang Zhang, showed how CRISPR-Cas9 could be applied for genome editing in mammalian cells. The lead author on that groundbreaking 2013 paper was Li Song, and we're delighted to have him with us today. Leo, welcome to the show, and thank you for joining us today.
Thank you both. Thank you, Jason. Thank you, Sanjay. It's great pleasure to be live at the conference today. Yes.
Before we dive in, we'd love to touch upon your journey in the field. From your background in engineering during your undergrad, working with George Church and then Feng Zhang, and your stint at Moderna, can you walk our listeners through some of the highlights of your journey that led to where you are today?
Thank you. Yes. Again, really exciting to be with both of you as live, in the conference. I've been coming here many years, thanks to the trainings, my mentors. But maybe to go back to the early days, I was electronic engineering student in college. Initially, want to be a game designer. That was the early passion. But when I saw the human genome project actually was completing when I was still a college student, that was incredibly exciting, and I thought to myself, maybe it's actually really cool. Instead of designing games, maybe there's a possibility to design genomes, design drugs. So I switched in the middle of my college to bioengineering, and that really led me to doing a PhD at Harvard. I joined the George Church lab and met a really amazing scientist, Feng Zhang, so I was co-advised by both George and Feng. So that was really incredible. Like Sanjay, we were just saying, amazing time, when Emmanuel, Jennifer really published this landmark paper, and then a lot of people, including us, were excited to try it in human cells and worked. And of course, opened a lot of exciting later studies and therapeutics as well. And after that, I thought, in addition, so the last training I did was Aviv Regev. She was a computational biologist at Broad. The reason for doing a postdoc with Aviv was kind of coming back a little bit to the original idea, right? We have a tool now, CRISPR, powerful tool. We also needed to understand how do we design, how do we target the genome, what to edit. And with Aviv, I was working on computational genomics, single cell genomics, to understand what are the target genes that we can engineer to build better therapies, cancer therapies. So this was kind of why I went to Aviv's group to do a computational training before starting my own lab at Stanford. Yeah.
Really cool, really cool. And Lei is also honored at this year's ASGCT meeting Outstanding New Investigator Award, and he's giving a lecture on his work at Stanford, AI-driven large gene insertion and functional genomics at the digital-physical interface. Obviously building upon that foundation and now applying it to such cutting-edge areas. But before we go there, maybe looking back, what do you think the field got right early when you're at that leading edge at Harvard and the Broad? And what do we fundamentally underestimate about the gene engineering tools that you helped develop?
Yeah, great question. Thank you, Jason. So maybe two really quick thoughts to share with everyone here. One is I really believe that the reason for CRISPR, of course, becoming so powerful and, like you said, have incredible early progress was thanks to both, of course, fundamental studies from Professor Doudna and Chametier, but also the zinc finger field. The entire genetic field really honestly didn't start with CRISPR. It really started with zinc finger proteins. Of course, many of my collaborators, like Dr. Matt Portis, was pioneers in that, but many others, of course, before CRISPR came, were able to use zinc finger, like Keith Zhang. A lot of people in the field were able to show that you can program a protein to engineer DNA in the genome, and that was kind of the blueprint, right, for how we could leverage CRISPR. Like, for example, through understanding the zinc finger, we appreciated how you can actually, basically target a gene and then perform different kinds of editing, like epigenetic editing, genetic editing, different types. So I think that's one critical reason that we were able to make so quick a lot of progress. But you mentioned that what we didn't appreciate is how long it takes, right? We were talking about 2012, right? The CRISPR landmark papers coming out, but the first CRISPR-based gene therapy was approved sometime in 2024. Really, just 2-3 years ago. Took more than 10 years, at least 10 years, right, before we see a first FDA-approved drug. And what I think people don't appreciate is when you have a new tool, you still needed a lot of time to understand the on-target, off-target, the toxicity, how do we deliver, right, these systems, including using some of the technologies you mentioned, from Moderna, from other places. So I think there's so much steps, right, from a initial, pretty much we have all the components, right, 2012, but then it took many steps, to really make a drug. So I think, of course, many of the work happened or presented here.
That's a really good point. I was lucky to, when I worked at Biogen, to collaborate with Sangamo Biosciences, applying zinc finger nucleases to reactivating fetal hemoglobin. And one thing that we learned during the development of that program was this on and off target profile. And obviously zinc finger nucleases, tremendously powerful, and you can have really great nucleases programmed to edit the genome on target, right, and have low off target. But if you do have a high off target, the burden to recreate a new system was so high. That's why when I saw the fundamental work that you and others did, and how quickly you could reprogram Cas9 or other CRISPR proteins to other new places in the genome to optimize that on-target versus off-target, right, profile, I think that's why so much excitement was raised behind this field. But as you spoke to, I think it still takes a long time to carefully understand those profiles in, right, a way to make sure you understand that risk-benefit profile. So thank you for highlighting that.
Yeah, I think, thank you for that insight, because I think CRISPR, when CRISPR was discovered, when you guys discovered CRISPR, right, I think the whole field was also evolving, right? Regulators were also understanding, kind of how this could be made into a drug that, right, could be approved and have curative potential. So the whole field was learning, and I think that's one of the reasons why it took so long. Right? Totally. Manufacturers were learning, regulators were learning, yes, and all of us were learning too in the same process. And
absolutely,
Jason and I were lucky to have been part of that those early days, both at CRISPR Therapeutics when they had that collaboration with Vertex to have Casgevy approved.
Yes,
and we were learning along with them. So, yes, it was really very. It's a long process, but we learned so much from it. Yes. And I hope it sets precedent for future CRISPR drugs, right? That would get to be faster in terms of approval. So your lab at Stanford is developing now cleavage-free gene insertion systems like SSAP, which is based on the microbial single-strand annealing proteins that's fused to dead Cas9 to enable DNA insertions. Yes. Can you briefly explain this technology in simple terms?
Yes. So maybe the simplest way to think about it is we all know Cre recombinase, mm-hmm, very powerful. We know that essentially it recombines two pieces of DNA, and if a patient is missing a key gene or have a mutation that inactivated a gene, mm-hmm, if we can use a recombinase, right, to essentially recombine a donor DNA into the genome, then you can give the patient or give the target cell a fresh, correct new copy of a gene as sort of a cure almost, right, for any errors or loss of function in genetic diseases. So we really thought, what if we have a CRI that can be targeted to anywhere in the genome, versus classically, as we all know, CRI recombinase have a single sequence that it will work. So only that sequence, of course, can be engineered a little bit, but essentially it's a very single site recombination protein. And because so that's I think a similar way to put it. And maybe the second quick point here is the motivation to work on it is CRISPR-Cas9 and of course zinc finger as well are amazing at inactivating a gene when we do a cutting, knocking out. Though a lot of diseases like we were just mentioning, loss of function, would really be not necessarily cutting a gene, but rather restore or insert a gene. So we thought that could be, and when I started my lab, I was thinking, okay, what's the next that could come that actually further move the scope or the limit, right, of the technology. So that's why we decided to work on thinking about a way to have a recombinase that can be targeted anywhere in the genome that's programmable. And that leads to the idea of using a dCas9. So doesn't cut, dCas9 is deactivated Cas9, right? Doesn't cut DNA, but it's like a GPS. So it can target a payload, through guide RNA to a particular location in the genome, like as you mentioned, very easy to program. And the payload would then be ideally a recombinase that is not tied to a specific sequence, but instead it will just do the work. So basically it will be essentially inserting a piece of DNA into the genome, and once the dCas9, right, bring that recombination protein to a specific site, it will work. And the simplest, at least at the time, the simply recombination protein that we know in nature, not surprisingly, is from the simplest organism, phage. So these SSAP or single-strand eating protein are what phage uses to sometimes inserting themselves into bacteria just to hide it, right? To hide themselves in the bacterial genome, or to repair, by recombining to phage DNA. Anyway, so we can go into some further detail, but broadly speaking, right, we found a simple recombination protein that from phage that could act as sort of the functional unit, and then the dCas9, thanks to CRISPR technology, can be the GPS that bring that functional unit anywhere, hopefully, to the genome. So that was the idea.
Oh, very cool, very cool. I love that your work is not only building upon all this foundation in understanding bacteria, but also viruses that infect bacteria. Yes. So very, very, very cool system.
Yeah. Do you see the system as a natural improvement over preceding technologies, or a genuine and necessary shift away from nuclease-based editing that relies on cutting of the DNA?
Oh, that's a great question. Maybe there might be three really cool things to talk together here, right? One, of course, because of the number one, I think there's a toolbox now, as we're discussing, starting from a single enzyme to a toolbox, and we're including seeing the power, like Sanjay, you mentioned, of non-cleavage type of editors, including, of course, I actually think the most exciting ones actually has been base editing, prime editing, and some of the related tools. I think that was incredibly powerful, and exactly as you said, they push a step further, not relying on the double-stranded break, or at least not really push beyond that. That's, I think, the first exciting thing is the toolbox is expanding and powerful, represented by base editing and prime editing. And second thought is, for our own tool, in a similar way, we believe, as we discussed, the potential of having a large gene insertion tool as a sort of a corrective gain of function type of technology, and we hope that the SSAP tool is one of the options for drug developers, for researchers, and that reason being, as we're just talking about Casgevy, right, the CRISPR therapy, took many years to figure out all the steps, and for one key step, which is delivery, in some cases, like in the ex vivo, right, it's easier or it's compatible with, for example, protein RNA delivery. Some in vivo settings, we need a different tool. So depending on the exact right format of the therapeutics, I believe we might need different types of systems. Like for example, SCCP usually, right, would need a DNA donor. But in some other large insertion tool, like the retrotransposon, some of the really cool tools, they might don't need a DNA donor, which could be beneficial or could be differentiated in terms of how you can deliver them. So that's the second. And the last point, I believe that there is just so much hidden treasure in nature. So I wouldn't be surprised there might be something that's a lot better coming in, maybe next year, maybe this year at the ASGCT sessions that we're gonna see. So there's always more maybe to come. So
Really cool. You mentioned delivery. In terms of limitations of your platform, do you see delivery as something so many people talk about for their editing technologies as a potential limitation and obviously an opportunity for innovation, but maybe taking delivery off the table, what else would you like to see, or how else do you think you can push this system further as you're looking ahead to applying it to hopefully correcting genetic disease?
Yes, I think that maybe one really specific point we've been working on, and hopefully we'll get a little bit of time to share actually during the presentation here on the conference that I will do, is the idea that similar to, as we know, in base editing, initially there was just adenosine base, like the ABE, but later through engineering, through evolution by David Liu, especially pioneered by Dr. Liu's group, we see this ability to engineer much more potent, much safer version of base editor. So we initially have these tools similar to other tools, or maybe 1.0 version. And what we recently have been doing is trying to leverage structure-guided design, AI-driven design, to understand the activity of these SSAP editors and trying to engineer mutations into these enzymes so that it will be high efficiency, less off target, and also more potent in terms of, for example, their activity to target specific sites. So there's really, I think, a lot of work that we could do to further enhance or improve over the 1.0 version.
So I would like to focus on your use of AI to drive innovation in your work. Your lab website provides a software, a section where visitors can see a menu of AI tools developed or used by your lab. Can you highlight a few of these areas and impact they are having on your lab?
Yeah, thank you. Maybe I'd like to quickly highlight 3 sort of AI tools or technologies we were excited about, and that kind of almost in some way present the evolution of my own group. And the first tool was a tool that one of our first graduate students in the lab, Nick Hughes, developed when he was a PhD student with us. So Nick was really interested in using AI to quickly design guide RNA. Essentially, we all know that sometimes in the early days, whenever I target a gene, EGFR, or P53, you would screen a number of guide RNA. And what Nick wanted to do is, if AI can learn from all the published genetic data, our own lab, other labs, it could learn which guide RNA design are more efficient, which guide RNA design less efficient. Of course, many people at the broad, in Wellcome Trust UK are all doing the similar work, but we had similar idea where we could train an AI model to understand the sequence determination of genetic efficiency. So Nick designed a tool called it's a very cool tool using a lot of data. Specifically, we're trying to design CRISPR-Cas12 guide RNA. So Cas9, many tools, but Cas12 is a very incredibly more compact, very interesting tool. A lot of companies are developing it. So we thought we should develop a tool for Cas12. And so we were able to develop one of the first, I think, tool, deep learning driven AI sort of software that help you to quickly rank different designs of guide RNA for a gene, and then hopefully, right, instead of screening 10, 20 guide, you can just use OR tool to find the top ranked 2 or 3 guide RNA, and hopefully, some or all of them would work. And of course, we experimentally tested it, and it works very well. Around, I think, at least 80% of the time.
In terms of both on-target as well as off-target efficacy?
So, great question. So we did optimize primarily for on-target, but also have a secondary sort of algorithm for off-target.
Okay.
But as we discussed, there needs to be data and knowledge first, right, for this AI to learn. So I think there are a lot more on-target versus off-target data. So we still need more. So that's first tool. I think very exciting at the time. And second tool is we built a AI agent called CRISPR GPT. So it's a very exciting sort of idea that we wanted to do is, what if we can train the AI to think and design experiments like hopefully a CRISPR domain expert. So what we did is we take about 11 years actually of discussion from actually a gene editing discussion forum where a lot of people from UC Berkeley, MIT, Europe, European sort of institutes are kind of just on the internet discussing gene editing experiments, what type of things worked for me, what didn't, and we took all these discussions, almost like how ChatGPT worked, right? By taking all the internet of text and train on it. We thought, what if we take a lot of notes, right, discussions from scientists in the CRISPR field to train we can call it CRISPR version of ChatGPT, and then the idea becomes, and the motivation really is very simple. If my new student, right, in the lab, or my new collaborator at Stanford or somewhere else came to us, say, I want to learn I'm a first year PhD, I want to learn how to design CRISPR, what is the protocol that's great for engineering T cells, right? In the past, it's more like, we have a senior lab member or myself, right? Train them or send them papers and read this and that, and have a meeting and training sessions, or come to ASGCT for the workshops. And now the idea is maybe there at least can be an accelerator where the AI can do a first, pass of almost like a sort of self-learning with the training, which speed up, right, the training process. And of course, when they hit some key questions, they can still come to a human expert. So that so one of the showcase in the paper we published last year on CRISPR GPT is that we let a collaborator who's studying a specific mutation in Alzheimer's disease, and they were doing, it's a human genetics lab, so they were doing CRISPR for the first time. So we actually let them use the CRISPR GPT to design the guide RNA, the protocols, and then of course our team helped to double check, and then they run the experiment using a CRISPR GPT designed protocol and guide, and it worked for the first, even the first time they tried it in their lab. So it led to a Neuron publication together with us. So it was amazing to see that we can hopefully have this tool help a lot of people.
That's such a really great example, and I love it because I've worked in both the stem cell field and now genetic editing field, and I think a lot of what happened in the early days of embryonic stem cells is you had all these stem cell scientists trying to apply it to areas like Alzheimer's and CNS, or I studied germ cell formation and how somatic cells make this decision. Do I stay a somatic cell or do I become a germ cell? And I think a lot of us struggled, and we really need to rely upon collaboration because we didn't have that content expertise. We had the stem cell foundation or the gene editing foundation, but we don't have the disease expertise. So it's amazing that you're developing a tool that can bring clinician scientists or people who have no background with CRISPR programming nucleotides, the DNA, and having that clinical knowledge of disease and what patients need in this field can only benefit and hopefully lead to faster therapies. So I like you also use the word acceleration, because obviously I think a lot of people are thinking about AI and how, is this going to take my job? Is this going to do a better job than I can as a scientist designing an experiment? I look forward to the day when the answer is like a resounding yes. It's like really cool and an opportunity for acceleration, but can also be a little threatening, I think, to people. So like on that side of, where do you think human intuition is really shining? Do you look at it as pure just acceleration, or do you think this is something that can really lead investigation?
It's a great question, and probably ongoing debate in some way in the field, right?
Fun to talk about, too.
Yes, very fun to talk. I would think this probably is also in the context of a broader sense of how AI is actually working or not working, right? So at least when we're testing a lot of these systems, for example, we definitely agree that because it was initial learning, right? We publish papers, but we don't necessarily publish all the knowledge from our lab or from our research team. So the first thing we observe is definitely if you have the AI system learning from human knowledge that's not in the public domain, you can make the ChatGPT a lot better. We do evaluations using benchmarks, using real experiments, and we know that when we have a specialized system like CRISPR-GPT, it would do better than ChatGPT. Though because it's still learning, right, from humans, experts, and sometimes hallucinates. So we are definitely not seeing, for example, a system like CRISPR-GPT would really be either doing things on their own or replacing human experts, but it is more really assistant. It's like essentially speeding things, especially for people who are new to the field, right? And I think that's almost like the same thing where, like, for example, I think a lot of people in my lab don't have coding training, but now you can really prompt these AI systems to build a website or web page, right, or build a little tool or do the data analysis. I think is speeding up. Not to say that the tool is already better than a bioinformatic scientist, but I think that's one thought first. And the second thought, very quickly, is that we are seeing the idea that because the AI systems can get feedback and can be run by many, many people, right? So you and I, the most efficient way for us to communicate probably is in the conference like this, ASGCT annual conference, where we can talk to a few thousand, even of course online, more people. But AI system can be used by millions of people theoretically, right? Because you can just build a website where people can log in. So if a lot of people are using it, giving the AI feedback, that could start to maybe make the AI system start to actually learn beyond what's published or what's available, because it's kind of learning from millions of scientists theoretically, right? Of course. So I definitely agree that there has been, at least in the community, people at least either hoping or claiming that the AI can become an AI scientist. Personally, I think that's why if we talk about some of the recent work we do with Lab OS, or we call it co-scientist, we still think that it's still not yet at the level where you can let it run by themselves because of the problem with hallucinations that we discussed, and a lot of times these digital systems are now grounded in the physical reality. Like you will believe, maybe I can do 10 million electroporation at the same time, but in reality, we don't have that ability to do it in a single lab. So I think there's grounding in physical reality is still a limitation for all these AI systems because they were trained in digitally, right? So for coding, it's great, but for actually running or thinking about a physical experiment, is still not there. At least when we were testing or evaluating it.
Really interesting. Really interesting. You spoke to this operational effectiveness side in the lab, where you are actively employing, I think, AI tools across, experimental design, which you touched upon, but also execution. It seems like data collection, which is obviously an area where I think a lot of scientists, particularly in the academic space, but including in industry, data documentation is so important, but often something that gets pushed aside to try to run that next experiment. Can you speak more about these efforts to employ AI for operational effectiveness in the lab?
Yes. So this related to some of recent work where we believe that it's almost like the Javis in Iron Man, where ideally, right, we're not just sitting on the computer all day. I'm sure most of you have been on the bench or working with teams, right, on the bench. we need to do actually make the drug happen in the physical labs.
Move the liquids around.
Exactly.
Move the liquids around.
Yeah. So we really thought it would be helpful to have this AI be able to at least help you to document, to actually execute, like just you said, execute some of these experiments. So what we did is we wanted to create a two-step process. First is what we call AI XR scientist, where basically you can connect the AI to cameras, sensors, equipments, right, in the lab to help you automatically record and capture some of the data that are critical factors, for them temperatures, right, volumes of things, incubation time, that are very helpful to know in order for us to be more intelligent on what factor can lead to a more successful outcome in the experiments. Cell culture density, all this stuff, right, that are, of course, human can record it, but sometimes it's almost like I wish when I'm running experiments, somebody else were standing by me, help me to make all the notes inside myself.
Yeah, that's a good dream.
So I think the first level is sort of an AI that help you, right, to record, understand, and maybe even remind you sometimes, right? So that's why I also have a smart glass version of this. But the second step, really, like we discussed, is execution. We know there's already a lot of liquid handling robots, for sequencing library preps, but we thought there needs to be a more flexible system. So we are definitely working on flexible automation systems where we can connect AI to robotic arms. And so learning alongside human scientists, we can start to see, there's a very repetitive step in the protocol, like shaking things or pipetting, repetitively. And if robotic arm can actually automate that, you can imagine this is almost like a ChatGPT, but with a physical arm, right? You can say, now that I need to, vortex and mixing this sample for 5 minutes, you just tell it or type it, and then it will automatically do it. But if the next day you want to do it, a different thing, like vortexing and then extracting something for 10 minutes, you can just simply type it. Instead of traditionally, right, the system will be more fixed. You build a particular robotic or liquid automation system, it will be good for one type of workflow. We wanted to build this AI robotic system where you could change the protocol every day or every month workflow, and it will actually always be similar setup, but intelligent enough to do different type of procedures.
Really cool.
Yeah.
That really hits home, because I think a lot of times I've faced that decision of, do I just brute force this, or do I take the time to try to reprogram? And typically I would always choose the brute force method, because I find it more rewarding as a bench scientist to go that route, I think. But so it's amazing to imagine AI tools to help sort of balance that,
Right. And on that, I have a quick question for both of you. Like, how much are you seeing, sort of automation happening in the physical labs these days, in this industry or across?
Yeah, I think it's something that all big and small companies are definitely thinking critically about. How do we do that? Obviously larger companies have the resources to explore that more, whereas currently I work at companies that are 50 people or less,
right?
And there you just have your critical path work streams, and I hate to use the word dabble, but dabbling with how AI can improve workflows. And like many people, I'm very naive and want to learn more about this. I think my entry to AI is like so many people. The first time I used it was to create a job description quickly, where it gets 90% right, and then you improve the other 10% for what you want. That's expanded now. A lot of regulatory submissions are somewhat routine, like study reports that have a lot of formal structure. So I see a lot of acceleration happening for incorporating AI, at that side for regulatory filings.
Yes.
In my exposure there is primarily Module 2 and Module 4, but I assume that's happening sort of throughout there. But I think you're really, the work you're doing is so exciting, because it's at this interface of therapeutic development, wet bench experiments happening in the lab, how to program these powerful editing technologies of the genome, and you're sort of capturing this across so many different areas, and it's why we were really excited to talk to you today. You mentioned going back to outlining sort of 3 areas of AI.
Yes.
Did this operational piece
Is the last.
is the last? Okay.
It's bring AI to the physical world, like you said.
Bring it
You
to the
know,
physical
we
world,
we are we're trying to make drugs and therapeutics, or at least accelerating that. I think eventually these drugs needs to be given to patients, not digitally, right? It has to be physically developed. Yes.
A really good point. You know, maybe you can touch upon that translational aspect of what you're doing and looking ahead.
Yes.
How you're thinking about translating what you're working on to the bench, from bench to bedside.
Yes, thank you. This is truly exciting to me because ultimately we hope that this will have real world impact and patient impact. So the to the question, I really believe in the power of working with clinicians, physicians, scientists, and this is what we are starting to actually be able to do. Of course, initially working with physicians at Stanford, very close to us. And one amazing collaborator we have is Dr. Mark Porteous. So, Dr. Porteous' group has been a pioneer in the field, but also, of course, importantly, considering how we could make these custom and tailored gene therapy that are curative and efficiently, built, tailored to a specific patient's mutation. And also, because his group pioneered hematopoietic stem cell editing, HSC editing, so, connected to Casgevy, I think he's actually a scientific co-founder of CRISPR Therapeutics as well, right? So I think that there is actually clear paths. Once, if we can, for example, tailor a CRISPR gene editing system that's optimized for a patient mutation, and for these HSC-driven disease or blood disorders, we could actually immediately apply in a translational way, because we know at least one case, right? In the case of anemia, SCD, sickle cell anemia, all the disease, the mutation can be corrected through gene editing hematopoietic stem cells, HSC stem, and then giving the corrected cell back to the patient, and then it's curative. And now, what is exciting to us is first, right, Dr. Porteous is trying to bring this Casgevy success, right, to a broader range of patients, especially for them, both people with inborn error of immune sort of systems or immune deficiency, SCID, right? The problem is a lot of these kids, and a lot of these are really, right, young, very young patients. Each of them have a little bit difference in their mutation, not like anemia. So all the problem, right, we have now is how can we quickly, right, have a CRISPR that work for that patient's mutation, and seen by also like success from baby KJ by Dr. Kira Musunuro, collaboration with Jennifer Doudna and a lot of people in the field. We are seeing this power. So with Dr. Porteous, we're thinking, how do we have an AI system? And we tentatively call it HTR GPT. The idea is that we want to build an AI system that can be optimally sort of trained to design CRISPR to do this homology directed repair. So this is kind of a repair-based gene editing that can correct mutation based on the specific patient's error, genetic error. And what we're seeing is that we can train with a lot of data, real data, from the Porteous lab over the last decade, where they have been doing a lot of this stem cell editing. And then we can train the AI to really know how to design a guide based on the specific patient mutation. And this is very cool because if you think about it, there's also the idea that you wanted to avoid off-target. So there's a lot of work, regulatory speaking, right, to make a design that will be safe and also effective, and then rounds of testing in the lab. But now, after training with all this data, of course we collected publicly and from Matt's group, we actually have run tests that if we just test 4 guide RNA, we can always find the top guide that are almost, over 50% effective in correcting, not just knocking out the gene, but correcting the mutation. So essentially, this cross the therapeutic threshold. So we think that essentially, instead of testing many guide RNA for any mutation using this HDR AI tool, we can actually only test at most 4 guide, hopefully, this in the set of genes. And finally, the cool thing is when we try to use the system, this is trained with a variety of stem cell data. We actually try to use the system for engineering what Jason mentioned, iPSC cells, so induced pluripotent stem cells, iPSC cells. It actually works really well and much better than a lot of the existing tools that are trained using cell lines. And we believe that, right, there's specific, right, cellular context in stem cells. So if you have a model that's specifically trained on the stem cell data, it will outperform a lot of existing tools, because existing tools usually are trained in cancer cell lines or some type of cell line. So we really now are seeing it's almost like if you wanted to have, for example, the best AI for designing job descriptions or writing regular document, you better first let it see in some examples or know how to prompt it. So we think that we're prompting these systems, right, with stem cell data, and that becomes very powerful, translational, and we're actually thinking that now that we might have a path towards using the tool, AI tool, design a small number of candidates, quickly test in the lab, very small scale, low cost test, and then you'll find the best candidate for hopefully directly trying to, made into a GMP ready, regulatory ready candidate that we can give to patient. In some cases, rare disease, as we know, there might be FDA accelerated paths for that. So thank you.
Yeah. Really interesting. One follow-up question there, and hopefully I'm understanding this correctly, but really exciting collaboration with Matt Porteous on the HDR side. In terms of that training with the 10 years of data, you mentioned the importance of cellular context.
Yes.
But how do you think those tools for maybe an HDR specific question, do those apply readily to like base editing, prime editing, or your SSAP tool? Or is each transition phase going to require new data sets to help refine?
Great question. Very quickly, you're totally right. For the time being, at least for the what we've tested, we need to get what we call in-context data for the AI to actually work. So base editing AI would need base editing data with sufficient amount. And this is totally true. And that's why we think that by, in some way, connected to what we were just talking about, data production is also so valuable, even for these digital AI tools. So if we have a better way, right, to better quality of data production in the wet lab through these AI co-scientists, through this AI automation, I think this will also help us and critical. So it's not just going to be training AI with existing data or a small amount of data, and then hope that it will work everywhere. It's really to have this end-to-end, like full cycle approach, right? We're considering AI, right, in the digital side, which sometimes really are helpful. We also need to let AI help us accelerate the physical data production, data quality, and then this turns into this full cycle, I think, virtuous cycle of improvements that hopefully drive a better gene cell therapy.
Yes, yeah, absolutely. I mean, and Jason and I were fortunate to work on sickle correction when we were at CRISPR Therapeutics,
yes.
And for HDR especially, there are all these rules, right? When how far the mutation that you want to correct is from the double-stranded break, the length of the homology arms, and
exactly.
that. So
and
it's
the
fascinating.
template design, like Jacob
Yes.
Thorne, of course, has published extensively on how he designs his homology template.
Single-stranded ODNs versus AAV,
Yeah.
as homology terms. So fascinating, and I'm really looking forward to these new wave of innovation through AI that you're going to bring. And I think we have just one more question before. If you're sitting here again in 10 years, what will feel primitive about today's gene editing approaches? What do you think will feel outdated?
Wow, this is a tough question. I think I tend to be optimistic about our field.
Yeah.
Though maybe to really specifically share thoughts on this question is, maybe 10 years later, we would really see two things that we probably are not going, not having today, and we probably think is too far today. But I really believe that maybe in 10 years, these two things can happen. Number one is that we will have very powerful AI systems. I think that can basically run experiments on digital and physical sort of environment. So the idea becomes, I think this is empowering. The idea basically becomes, right now, for example, a student run small number of experiments, and for the professors, the PIs, overseeing 10 students running 10 experiments. And similar in pharma, right? Your team, you have 10 people in the team, and they're running 10 people-ish worth of work. But if every member of your team have this AI assistant, right, running experiments, maybe even on the evenings, designing something, checking the data, it empowers you to have 10 people on your team, but it will hopefully have a productivity of almost 100 people today. So I think the scaling, what we call a scaling effect of these AI tools could happen across digital and physical world in 10 years. That's really my hope. And because that enable everyone to, drug development is so tough and long duration, so that I think that's helpful. And I'm hopeful that will happen. And the second thing is, as we were talking about, there's so many individual sort of knowledge that's still needed, right? We're still, in some way, right, seeing the early progress of this whole gene editing, precision medicine field, or customized, right, therapeutics field. I think in 10 years, I think we will see really this becomes routine, rather than one, baby, one patient, or one rare disease. I think this will be hopefully routine. We're making progress on delivery. You mentioned all these amazing presentations here at ASGCT conference. I think seeing all this progress, I'm very hopeful that we will have routine, these incredibly powerful gene editing therapies or gene therapies for not just rare disease, but hopefully common disease. I think this is going to be exciting. Thank you.
All right, look forward to that. Well, thank you so much for taking time out of your very busy schedule. Another follow-up question, you said you started with game design. You have time to play games in your busy schedule? Is that hard between running the lab,
right,
travel, kids?
Well, it's a great question. Usually, of course, on my way actually to the airport, coming to ASGCT, I was playing a little bit of cell phone games. I think that was the game that I'm playing.
Yes, still I do. That's great to hear.
Yeah.
Everyone needs hobbies.
Yes.
So,
Thank you so much. It's really good to hear from you today. Thank you for joining us. I look forward to your talk tomorrow.
Thank you.
And thanks again for all your insights. It's and for your contributions to the field.
Really appreciate
truly
the
amazing.
thoughtful questions and your time. Thank you.
Thank you. Pleasure meeting you.